And in a nutshell, I would like to say this about myself, that before the Lord called me to do this for Christ, to serve Him in this way, the Lord gave me an opportunity, an opening. And at that time, I was studying in the university here in Kenya. And He gave me an opening to go do a master's degree, a second degree in Germany. And this was under the German Academic Exchange Program. And so the Lord helped me so much. In the program I was in, I was the lead student in that program. So then I got a scholarship. And it opened a big door for me to go to Germany and do the things I could not do here. I was so much interested in genetics. I always wanted to know inheritance and how genetics affects life. And then right from when I had gotten the scholarship and I was about to leave for Germany is when all of a sudden a professor arrived from Israel. A professor arrived from Israel. His name is Professor Sam Mendlinger. And when he arrived from Israel, he came to me, he said the department, the faculty, had recommended me to him because he came looking for a student that can take a scholarship to Israel. And so at that time, it was amazing because in one of the hotels next to the Israeli embassy, actually, where he invited me to chat with him on this opening, when I sat with him and he said he would have loved to have me do my advanced degrees in Israel and work in his laboratory. And so it did not make much sense, though, at the time, because Germany was offering a bigger stipend. And you know, as a student at that level in Africa here, that's a key issue. How much stipend am I going to receive? Will I be able to receive enough to upkeep and help home, help my family? Will I be able to send something for them, you know?
Wow.
Yeah, to sustain them until one day I'm able to start working. So you saw that there was a lot more stipend offered by the German scholarship, and then the Israeli one was giving less. But for some reason I said, no, I am going to Israel. Because then I was in a place where I had to choose between the two. So I chose saying, I'm going to Israel, and I would like to go and see the land of the Bible. That's what I said.
You were not Christian at that time.
I was Christian, but my Christianity was not that very strong.
Okay.
It was in the superficial definition of Christianity you see today. I was not so much anchored into the workings and the walking and the lifestyle. But I knew that I'd received the Lord. But like you see Christians today wavering left and right. So that was the kind of salvation I thought I had. And so all of a sudden I made this irrational decision. Irrational. To go to Israel. And that's when I went to Israel. I abandoned the program for Germany, which was supposed to take me to Berlin. But I now went to Israel and I went to Beersheba.
Wow.
Beersheba, if you know how Israel sits, is within the Negev desert. Yes, I went there, interested in genetics still, inheritance. And when I finished from there, and then I got an opening to do a PhD. To do a PhD now, the third degree, in Haifa, moving north now.
Yes.
Haifa by the Mediterranean Sea. Yes, so when I went to do my PhD at the University of Haifa, at that place, I was interested in still the genetics, but now I had to, because in the master's program, what I was addressing was essential biochemical genetics, looking at enzyme systems, their isotypes, their variants, their inheritance, their value. But now, at Haifa, I was now able to squarely focus on molecular genetics, to look at DNA works, DNA structure. And if you remember very well, that was around the same time when there was a lot of investment globally.
Yes.
When the world scientific community had invested into the Human Genome Project.
Exactly.
So then the work on DNA was novel. It is now still, but I'm saying at that time it was now this big gold mine that was set in front of us. And so, and see, most of that work still, even as I went into the PhD, was really basic, absolutely basic, because the issues that I was interested in, the challenge that I was interested in to examine was the structure of DNA. What is the composition of the sequences, DNA sequences? And in trying to elucidate DNA sequences, to open up DNA sequences, we saw different segments, different zones. And the issue was, what are the relevance? You see, what is the importance, for example, of this ATG or GC, whatever, repeats on gene expression? You see, the ultimate is gene expression.
Yes.
How is the regulation of the expression of genes? So that then had pharmaceutical value. That then was now important. It can be used. It can be used to address real felt problems. So some of the techniques that were on the ground were really so primitive, I can't say so at this moment, because like rapids, which are like random amplified polymorphic DNAs. So polymorphism, you know, the study of DNA then was essentially based on polymorphism, meaning differences. So in looking at different regions, and another region, but then how do they translate into expression, into gene expression, and most importantly, how do they control the expression of genes? That one can be used in addressing problems. And so at that time, I was interested in looking at a certain type of virus, which unfortunately then was a European virus. So again, we have transnational quarantine. You cannot import them in your laboratory. So then I got involved in doing some work in Germany eventually.
Wow.
Also still within the DNA project, and looking at DNA repeats. And I want to mention this, I don't want to go very deep into this, but in studying DNA, we were able to find that there are certain random repeats of DNA sequences. The value we did not know.
Yeah.
We did not know what they stand for. And then we had now the coding sequences, now the sequences that define the genes of interest. But only later. So polymorphism was the issue, to say that what is the difference between this human being and this person, or between this animal and this animal, or this plant, you know, differences.
Yes.
Where there is a locus, base pair, they call them in base pair. There is one locus here, locus meaning where genes reside.
Yes.
One locus here and is absent here. So that is essentially key, because that is what is used in, for example, if there's a crime scene.
Yes.
If you want to come into a crime scene and you want to know who was at this site, so you use polymorphism. That's what you call the fingerprinting. So you see that this person has this pattern.
Yes.
And this person has this pattern. They will share a lot of genes that you see in the human genome. But then you will now find the difference in this person and this person.
Yes.
That is what defines this person from this person. So literally everything is based on polymorphism. Even the disease works. It's based on, if the patients that report, for example, in a particular clinic, I'm going to mention that in a while, have this type of gene, which has this sequence. And the patients that show this resistance to this disease, or tolerance, or are not afflicted by this, don't have that locus. Then you can now use that for screening. So this was the basic work we were doing in Israel at that time. And that's what my PhD was essentially based on. And then, eventually, now I moved on from there. I was so blessed at that time. It was when I was in Israel.
Wow.
It was really when I was in Israel towards the end of my PhD that one time in the night, the Lord visited with me in the night, and he called me 3 times by voice. And I remember that when he did that, I was so shocked that I woke up. And he left a message in my heart that I have called you.
Wow.
But you know, all that could not make sense.
Yeah, that's exactly. You come from a medical background
Yeah, and then in Israel,
and then you receive this calling that says, I'm calling you for this. How?
and then in Israel, you rightfully said, in Israel, Sundays is a working day.
Exactly.
You see that? So even the concept of going to church to try to harness this whole thing was almost not there. There were churches, there's those people that on Sundays, you know, Saturday really.
Yeah, Saturday.
On Saturday,
Yes, that's
on Shabbat,
Yeah,
when,
Shabbat,
when these forgot the synagogue, they try to hold the church, you know. But had not hooked so much onto the churches. And, excuse me, and so the other thing also, the Lord made me know that if I said yes to the calling, all the dream of being a professor in the university was over.
That was your dream. Your dream as a child was to
Yes,
teach,
my longing really.
my young dream.
To one day come back to Kenya and lecture in the university.
That was always in your heart to come back to Kenya.
Yes, and lecture.
Uh-huh.
Or wherever I go to, but at least lecture,
Yes.
be an academic. So this is something I was deliberately pursuing.
Wow.
You see that?
Wow.
And so the problem then became, the Lord made it very clear that if you agree to this calling, that dream is finished. You will not lecture. And so that confused me. So I did not understand. Then what is this all about? Because how will I even go back home and say, now is what, everything is over? I just want to preach the gospel. And so, and then, as I was finishing my PhD in Israel, then the attempt was to run away from the Lord. That's when I began out what I attempted to run away. Because he made me know that if you will go into this, forget everything else. Just come and serve me fully. So the first thing to do was to run away. To say, I don't want this thing, shut my ear, just move on with what I'm pursuing. Because it will not make sense.
How long you were in Israel?
In Israel, that's a total, the PhD was 5 years, but I'd spent 2 years in Beersheba. So that's a total of 7 years.
7 years.
7 years in Israel.
And in those 7 years, when the Lord called you? Year 6, year 7?
In fact, towards between the 6th and 7th year, towards the end of the PhD,
Wow.
is when the Lord, is when the voice called. And, you know, with all that, nobody to give you counsel, to help you. And, you know, let me tell you, one thing I really learned in that is that church should set up structures to help people. When the Lord calls them, to help them harness.
Yes.
Not even that, to understand the calling. You see that?
Yes.
At least someone they can talk to and share. And then tell them, oh, if that is what has happened, then begin doing this and that, you know. And so, but when I was eventually leaving Israel, I was, the Lord, he was blessing me in his own way, in a very mighty way. I received an invitation. Before I finished, actually it was the last 1 year, which was very unusual though. I received an appointment actually to a place called CABM in New Jersey. That's the Center for Advanced Biotechnology and Medicine. It was part of the UMDNJ, the University of Medicine and Dentistry of New Jersey. And so that was one. And the other one was Center for Advanced Center for Pharmaceutical Biotechnology. So you see, somehow things were working well. And for me, I did not understand that. For me, it meant run away further.
Yes.
Now pursue your things and don't worry about this now.
Well,
because it's working this way now.
Completely different change. I mean, you left Israel, go to the United States where it's a completely different lifestyle, completely different atmosphere. You thought that you were running from God. You thought that you were safe now.
Yes, because actually the calling became remote. Because, when you finish your PhD and then you go into environment where you start work.
Yes.
You see, you go into postdoctoral fellowship, you go into lecturing and all this. And then you get engrossed. I became deeply absorbed now. And the work was exciting. I mean, I thought this was also good for the Lord.
Wow.
Because looking at cancer,
Yes.
different drugs, especially trying to design the future drugs.
Wow.
You see, because I went into an area which is called signal transduction. Signal transduction is essentially drug metabolism, which means understanding when I take this medication, what are the biochemical pathways that are fired up in the body. And then in that way we are able to develop drug targets. You see that? So the team I was working with essentially, by the way, I decided to go to Chicago instead of going, first of all, to UMD&J in New Jersey, University of Medicine and Dentistry. I went to the Center for Advanced Biotechnology. Pharmaceutical biotechnology, sorry. At that place, we began to look at the future drugs. And essentially it was a process because I delved into signal transduction, which is part of drug metabolism, but hence I now specialized in oncoprotein networks.
Wow.
So which means I became so much interested in knowing the network of proteins that are important in oncology, and then how, let me just say what they are. Yeah. They are the mitogen activated protein kinases. We used to call them MAP kinases. And we found out that they were the novel drug targets for the future. Because they are the proteins that actually transduce, signal transduction, transmit the signals. You see that? Wow. When, for example, someone is on chemotherapy, so really my interest was in chemotherapy more than anything else. Yeah. So looking at different chemotherapeutic drugs that we're examining and the pathways they were firing up. So our main objective then, allow me just say this way, was to essentially exploit, there are basically two pathways. There's one pathway that is used for survival. We later we named it defense, body defense. It's like when you walk out there and someone is smoking, or you are subjected to what we used to call environmental insults, or you at your workplace exposed to mercury, whatever it is. There is a pathway in your body that regroups very fast, becomes recruited, so to say, and begins to detox, to save the body. So that was a pathway that we were interested in also. And they have certain oncoproteins that define the elements for transduction, the proteins that transmit. You see that?
Yes.
To the nucleus, from the cell surface to the nucleus. And then we also had now what essentially became known as the P38 pathway, really. That's the name. The P38 pathway is essentially the cell death. We used to call it the death pathway. So this is the pathway that most of the time, when the pharmaceutical companies are designing their cancer chemotherapeutic drugs, they exploit this. Because this pathway, once it's fired up, once you develop a product that will activate it, then it leads to cell death. In fact, we used to call it apoptosis, and meaning self-suicide. You are telling the cell, the drug is telling the cell, kill yourself. Wow. So it was a very novel thing to do. Wow. So you see, when I dived into all this and we were doing this, I thought that this was the most important thing to do. Because this is touching on real life. You see what I'm saying?
Yes. You're developing something that can save lives, something that can help humankind.
Absolutely.
And that was a great and novel thing to do also.
Absolutely. And a lot of investment, like you see in the US, from NIH, from the National Cancer Institute, the NCI also. And for me, this was very key. And then after that, I moved on from there, moved on to New Jersey. I eventually went to New Jersey. And so went to the College of Pharmacy of Rutgers, went into a group, the pharmacokinetics group, also dealing with cancer chemotherapeutic drugs, But then we were able to look at different areas, and then I moved on finally to the CINJ. I was actually appointed as research teaching specialists at that time. They used to call it RTS, RTS3,
Yeah.
research teaching specialists. But now, this is the big department of surgery, and then the division of oncology.
Oncology.
So it's surgical oncology
Wow.
at the Cancer Institute of New Jersey, CINJ. And there, the main work now was to look at, now, with a group of surgeons. You see that? This was very novel because now we had material right from the operation theaters, you know. And the key area there was tumor angiogenesis. How do cancers spread in the body? How do they eventually go through into metastasis, where now they are resistant?
Exactly.
And what genes do they exploit? And so I was very touched by the fact that we were able to find out, you see that, clearly that cancers actually use, through the process of angiogenesis, what they essentially do, I can summarize this. It's normally what cancer cells do, when a cell becomes malignant or becomes cancerous, what it does, it actually sends a chemical messenger. They normally send chemical messengers to the surrounding cells. And they tell the surrounding tissue cells, look, you begin to tubulate, meaning you begin to grow and become like a tube and grow towards me. So they send chemical messengers with instruction to the surrounding cells, telling those cells, please begin to grow but tubulate. That's essentially the process of angiogenesis.
Wow. Wow.
Tubulate and grow towards me and feed me. And then they feed sometimes sucrose and so forth. And the cancers, the tumors begin to grow. That's essentially the process they use. But now, having known that that's the process they use, then the challenge now from the research point of view became, how do we shut down that process? Do you understand now how it was important?
How it works.
It's always important
Exactly.
to understand the mechanistics, the basic preclinical. You see that? And then now, how do you shut down the growth of cancer? So we found out that these messengers that the cancer cells were sending, they were the vascular endothelial growth factor, which we termed VEGF, VEGF A and B. So once we found out that they were sending the messenger called vascular endothelial growth factor, which is essentially a very important thing. You know, angiogenesis is so important in the growth of teeth, whatever, the gums. It's a normal process in the body.
Yes.
But the cancers are exploiting it, you know. So now we found out there were two types, VEGF A and B. So that was key now from mitigation, for intervention.
Yeah.
Because now what we could do is very simple using mutagenesis, you know, which is the process of causing mutation in DNA. We could now be able to mutate the genes that encode for them in the cancers. So we have anti-VEGF A and B. So once you have anti-VEGF A and B, then it can shut down the messenger. And so these were the processes we were involved in. And when the Lord relentlessly,
He keep calling you at that time?
relentlessly from Chicago, even before I moved to New Jersey, again, in a very major visitation in the apartment where I was staying in a place called Forest Park in Chicago. Then one night in a major vision, then the cloud came
In the room.
in the room in a vision and spoke with me from there again to let me know that He has called me. And so you can almost see that when you follow the entire process, you see that I was not understanding.
Yeah.
I was really not understanding who was calling me,
Yeah.
the
didn't have
enormity of Him.
No specifics in the calling. He didn't say, this is what I want you to do. He just said, I just calling you, or He gave you some instruction of what, did you be able to see what you're doing today, or no? Was something just really vague?
That's very powerful. That's a very powerful question. Because the thing that really made me try to run away further is the fact that in the calling, He showed me what I'm doing today. He showed me what I would be doing. Essentially what we're having now going on today.
Yeah.
For example, you're in a big meeting and there are this huge multitude of people and you're praying for them. And I remember in one of those visitations, then His hand, the hand of the Lord, from the bed where I was lying, then the hand pointed and said, go pray for my people. So I could see, he said, pointing at the meeting. And when I looked at the people, I saw myself preaching there. So I saw myself already then.
Wow.
Yeah, I saw the future then.
You were there. Did you be able to see healings like you're seeing today? Or?
Tremendous. Tremendous. And that is now what complicated the whole thing.
Yeah, because if as a medical researcher, PhD with all these titles and knowledge, let's take the Christian approach and let's concentrate on the medical approach for a minute. Mm-hmm. How was to see, as you describing cancer, describing all these things, how was for you to see, for example, all these diseases like AIDS and all these, you saw that in a dream, in a vision. How was, that was the most difficult thing for you to understand, I guess.
That's very powerful. That's very powerful that you say that. Let me tell you, Pastor Gamboa, now, that is what complicated, to me then, complicated the calling. Because, for example, I remember one time, in one big dream, he showed me the carpet, the carpet that was laid, the carpets, you see them laying today.
Yes.
And then I could not understand, of course. And the people are walking. A cripple was walking on the carpet. And I saw this particular case where someone had a cancer and a tumor and it burst, Yeah. But now how did that complicate the calling? Because I asked myself, how do you come from this dream and go tell colleagues and go tell people that you're working with, people who are looking for evidence?
Exactly.
In fact, they are trained to take evidence. Mm-hmm. That's their training. Mm-hmm. And tell them, look, I see tumors dissolving in the Lord's meeting. Of course, definitely, you would be written off, or they would say you are crazy, something's wrong. You see that? Mm-hmm. You need help, And so that is now what complicated the calling.
But do you doubt it? Do you question that coming from your background, or you believe in that?
Well, let me tell you one thing. When the Lord spoke with me, I knew it was going to happen. Because it's so vivid. I've lived it though.
Yes.
I've already lived it. I've seen it, and it's very mighty, and it was absolutely enormous. Even overwhelming. To handle it was difficult. I remember one time I said, "Let me try to put these things in writing," but then it was even difficult to write.
Yes.
Because you cannot describe. You don't have any mechanism. You cannot write. You know. But anyhow, that is what complicated, because I did not know how to go out and tell people and tell them, look, the Lord is showing me that I'm praying for people, and now the cancers for whose cure you're looking are being healed. So, to not have made sense. And that's what complicated the whole thing, until eventually I finally accepted the call of the Lord. But it was a process. I'm just trying to summarize the whole thing.
Yes,
it was a process, really, because I was then looking for pastors. At one point, I began to look for pastors.
For answers.
No, pastors. To sit with them.
Yes.
To tell them what I'm seeing, what I'm hearing is saying. Because the